Welcome to this week's lecture on animal renal systems, water and waste. Life originated and diversified in a watery environment. Consequently, the chemical functions of cells and organisms crucially depends on the property of water and the relative amount of water and electrolytes inside a cell. A healthy person can live several weeks without food, but cannot survive much more than a few days without water. Homeostasis of water and electrolyte concentration is critical to normal physiological function, not just in humans, but in all organisms. When there is a difference in water concentration between two regions, a net movement of water molecules occurs from regions of higher water concentration to regions of lower water concentration. This process is called osmosis. Water often contains other dissolved molecules such as electrolytes, amino acids, and sugars, which are collectively called solutes. The ability to control osmosis, and therefore to regulate levels of water and electrolytes in cells, depends on the property of their plasma membranes and embedded proteins. When a membranous barrier allows water or solutes to diffuse freely, it is said to be permeable. When it blocks the diffusion of water or solutes entirely, the membrane is considered impermeable. When it allows the movement of some molecules but not others, it is semipermeable or selectively permeable. The membranes of cells are selectively permeable because of the combination of the lipid bilayer and transmembrane proteins. Plasma membranes and sheets of cells that are semipermeable allow water and smaller solutes to diffuse freely, but prevent larger solutes, like proteins, from passing through them. Water is able to move directly through lipid bilayers by diffusion, or through channels called aquaporins by facilitated diffusion. Osmotic pressure is the pressure needed to prevent water from moving across a selectively permeable membrane. It reflects the tendency of water to move from one solution into another by osmosis. The higher the solution concentration, the higher the osmotic pressure of that solution, i.e., there's a greater tendency for water to move into that solution. We can see this indicated here by the larger arrow showing water movement toward the solute compared to the smaller arrow flowing back toward the solute-free side. Whether water moves by osmosis into or out of a cell depends on the solute concentration of the inside of the cell relative to the outside. Water movement is driven by difference in total solute concentration between the two sides of a cell membrane. Water moves across selectively permeable membranes in response to concentration differences of any solute. That could be electrolytes, such as sodium or potassium, or another dissolved molecule, such as glucose, or an amino acid, or even waste. Osmoregulation is a form of homeostasis and is the regulation of osmotic pressure or water content. It keeps internal fluids from becoming too concentrated, that is, high osmotic pressure, or too dilute, low osmotic pressure. High osmotic pressures inside a cell can damage the cell, sometimes even causing it to burst, and thus disrupting some of the animal's functions. On the other hand, excessive dehydration impairs a cell's metabolic function. To regulate water and solute levels, and hence the osmotic pressure inside cells, the cell controls the solute concentration of the inside of the cell relative to the solute concentration outside of the cell. At the level of the cell, osmoregulation is achieved by the movement of solutes, particularly electrolytes. At the level of the organism, osmoregulation is achieved by balancing input and output of water and electrolytes. Water can be gained and lost in different ways. In general, animals gain water by drinking water that is less concentrated in solutes than their body fluids. Water is also acquired through the food that animals eat. Additionally, animals produce water as a product of cellular respiration. Electrolytes are also taken in and lost in different ways. Animals gain electrolytes in the food they eat. If the water they drink has a higher solute concentration than their body fluids, animals can gain electrolytes by drinking. Humans lose electrolytes as well as water when they sweat. Electrolytes are also lost by specialized glands and in urine and feces. The ability of many organisms to regulate their water and electrolyte levels has enabled them to maintain homeostasis and to live in diverse environments. Animals regulate their internal osmotic pressure in different ways. Some animals match their internal osmotic pressure to that of their external environment. These animals are osmoconformers. They keep their internal fluids at the same osmotic pressure as the surrounding environment, which reduces the movement of water and solutes into or out of their bodies. They don't have to spend a lot of energy regulating osmotic pressure. However, they have to adapt to the solute concentration of their external environment, which is typically seawater. Most marine invertebrates, such as sea stars and lobsters, are osmoconformers that match their total intracellular solute concentration to the solute concentration of seawater. Some marine vertebrates, such as lampreys and sharks, are also osmoconformers. Many animals maintain internal solute concentrations that differ from that of their environment. These animals are osmoregulators. They expend considerable energy pumping ions across cell membranes in order to regulate the movement of water into or out of their bodies. The ability to regulate osmotic pressure allows osmoregulators to live in diverse environments: saltwater, freshwater, and land. Teleosts or bony fish, as well as all freshwater and terrestrial animals, are osmoregulators. These animals have evolved a variety of specializations for water and electrolyte balance and waste excretion. Many marine teleosts maintain concentrations of electrolytes much lower than the surrounding seawater. On the other hand, freshwater fishes and amphibians maintain concentrations that are higher than those of their aquatic environment. Aquatic animals use their gills to breathe air as well as to regulate electrolyte levels. The thin gill filaments, with their extensive surface area, are well adapted for gas exchange and also facilitate electrolyte transport. In both cases, the gills maintain a substantial difference in solute concentration between the fish's internal and external environment. A variety of animals have developed specialized mechanisms for releasing excess salt from their systems. Sharks and rays excrete excess salt by a rectal gland, which eliminates salt with their digestive wastes. Similarly, many marine birds have evolved specialized nasal salt glands to rid themselves of the high salt content of their diet. These salt glands work by actively pumping ions from the blood into cells that make up the gland, followed by excretion of salts from the body through the gland and the nasal cavity. Their salt glands allow these animals to gain net water intake by drinking seawater. In contrast, humans and many other non-marine adapted animals are incapable of deriving useful net water gain by drinking seawater because they cannot eliminate salt in high enough concentration. Excretion is the elimination of waste products and toxic compounds from the body. Most cellular animals have evolved specialized excretory organs, such as the vertebrate kidney, to isolate, store, and eliminate toxic compounds and metabolic waste products. When proteins and nucleic acids are broken down by metabolism, as shown in this figure, one of the byproducts is ammonia. Ammonia contains nitrogen and is toxic to organisms, so it is a form of nitrogenous waste. Ammonia can disturb the pH balance of cells and cause damage to neurons. At high levels, it is lethal to organisms, so it either needs to be eliminated from the body or converted to a less toxic form. Some organisms, such as fish, are able to excrete ammonia directly into the surrounding water, primarily through their gills. As a result, ammonia does not accumulate to toxic levels in these organisms. The movement of animals from water to land posed many challenges, among them the problem of waste elimination. Organisms evolved biochemical pathways to convert ammonia to less toxic forms, including urea. Because urea is less toxic than ammonia, it can be stored in a fairly concentrated form before being excreted. In mammals, urea is produced in the liver and is then carried by the blood to the kidneys, where it is eliminated. Urea is less toxic than ammonia, but requires energy to produce it and water to eliminate it. Ammonia can also be converted into uric acid. Uric acid is the least toxic of the three forms of nitrogenous waste, so animals can store it at a higher concentration than urea. birds and many reptiles, arthropods, and land snails excrete nitrogenous waste in the form of uric acid. Uric acid precipitates from solution and forms a semi-solid paste, familiar to many as white bird droppings. Because uric acid is not dissolved in water, it does not exert osmotic pressure and is eliminated with minimal water loss for the animal. This allows many reptiles and insects to live in extremely hot and dry environments. However, uric acid is energetically expensive to produce. Filtration, reabsorption, and secretion are the three steps that all animal excretory systems perform to eliminate toxic wastes, retain key electrolytes and solutes, and regulate water volume. Filtration is the process by which the blood is filtered into an extracellular space, allowing some substances to pass through but retaining others. In this energetically inexpensive process, circulatory pressure pushes fluid containing the wastes through specialized filters into an extracellular space. The filtered fluid, called the filtrate, contains waste products along with water, electrolytes, and other solutes, which drain into excretory tubules that connect to the outside. The excretory filter, like a cell's plasma membrane, is selectively permeable, allowing smaller ions and solutes to pass into the tubule lumen along with water, but it retains larger solutes such as proteins and cells. Filtration is followed by reabsorption, the process by which essential ions and solutes are transported back into the blood. Reabsorption is an active process for some molecules and a passive one for others. In both cases, substances that are important for the animal to retain, such as electrolytes, amino acids, vitamins, and simple sugars, are taken up by cells of the excretory tubule and returned to the bloodstream. Water is reabsorbed into the cells by osmosis, driven by the osmotic pressure established by the active transport of solutes into the cell. The final step in waste removal is the secretion of additional toxic compounds and excess ions. Like reabsorption, secretion is also an active process and involves eliminating substances that were not filtered from the blood earlier. Secretion is specific but energetically very expensive. The processes of filtration, reabsorption, and secretion serve two important and complementary functions. They allow waste to be isolated and subsequently removed from the body, and they allow the organism to adjust the amounts of water and electrolytes required to meet its osmoregulatory needs. To accomplish these functions, animals have evolved different excretory organs. For example, in flatworms, excretion involves isolating the fluid from the body cavity in excretory organs called protonephridia. This fluid isolation is done prior to filtering. Cells with beating cilia move the fluid down the lumen of the tubule to an excretory pore. Freshwater flatworms, such as Planaria, have an extensive network of tubules that eliminate excess fluid along with waste products. In segmented annelid worms, such as earthworms, the body fluid is filtered through small capillaries into a pair of excretory organs called metanephridia within each body segment. The fluid enters a metanephridium at a funnel-shaped opening surrounded by cilia. It drains the body fluid into a series of tubes in the next body segment and then out of the animal through an excretory pore. Blood vessels and cells surrounding the tubules reabsorb and secrete additional compounds as urine is formed. Like freshwater flatworms, earthworms reabsorb electrolytes and eliminate dilute urine containing dissolved nitrogenous wastes. In insects and other terrestrial arthropods, fluid passes from the main body cavity into a series of tubes called Malpighian tubules that empty into the hindgut. The cells lining these tubules actively secrete uric acid and excess electrolytes into the lumen of the tubules while retaining key solutes. Muscle fibers in the walls of the tubules help push the fluid to the hindgut. In the hindgut, the fluid becomes increasingly acidic because of the buildup of uric acid, causing uric acid to come out of solution when it enters the rectum. Because uric acid is removed from the fluid, it does not influence the movement of water. Water is therefore free to diffuse back into the tissues as electrolytes are reabsorbed from the rectum. The excretory organs of vertebrates are the kidneys. Like all excretory organs, they not only serve to eliminate wastes, but also play an important role in water and electrolyte balance. Vertebrate kidneys reflect the evolutionary transition of vertebrates from an aquatic to a terrestrial environment, where there is a need to minimize water loss. Mammals and birds, for example, can excrete urine that is more concentrated than their blood and tissues. The kidneys lie posterior to the abdominal cavity. Whereas the kidneys of humans and other mammals are bean-shaped, the kidneys of birds and other vertebrate animals are elongated and segmented. Paired vertebrate kidneys receive blood supply from the heart by renal arteries that branch from the abdominal aorta. Vertebrates filter their blood through specialized capillaries that have openings in their walls. These porous capillaries form a tufted loop called a glomerulus. There are thousands to millions of glomeruli in each kidney. Driven by circulatory pressure, nitrogenous wastes, electrolytes, other small solutes, and water move through small holes in the capillary walls into an extracellular space surrounded by a capsule. The filtrate then moves through a series of renal tubules, which further process the filtrate by reabsorption and secretion before it enters the collecting ducts as urine. The collecting ducts converge on a larger tube called the ureter, which brings urine from the kidneys to a hollow organ called the bladder in mammals and fishes, or the cloaca in the case of amphibians, reptiles, and birds, for storage and elimination from the body. The glomerulus, capsule, renal tubules, and collecting ducts make up a nephron, which is the functional unit of the kidney. Nephrons perform the three basic steps of excretion and osmoregulation: filtration of blood passing through the glomerulus, reabsorption from the renal tubule back to the bloodstream of key electrolytes and solutes, and secretion of additional wastes by the renal tubules. The paired kidneys of mammals are organized into two layers: an outer layer called the cortex, and a deeper layer surrounded by the cortex called the medulla. Nephrons are organized into wedge-shaped renal pyramids. Each nephron's glomerulus is located within the renal cortex, or outer layer of the medulla, and feeds into a renal tubule that starts in the renal cortex before dipping into the medulla at the base of the renal pyramid, and then looping back up to the cortex. The collecting ducts of the renal tubules also descend into the kidney base, emptying into the renal pelvis and draining through the ureter into the bladder. The glomerulus is a tuft of capillaries with blood entering by an afferent or toward arteriole and leaving by an efferent or away arteriole. The tuft of capillaries is encased in a membranous sac called Bowman's capsule, and the space enclosed by the capsule is called Bowman's space. Blood is filtered as it passes through the tuft of capillaries. The filtrate passes out of the capillaries through Bowman's capsule and into Bowman's space. Water, wastes, and solutes from the blood pass through the capillary wall into Bowman's space before moving into the renal tubule. To allow filtration of blood, the capillaries of the glomerulus have a different structure from that of capillaries elsewhere in the body. The filtration barrier of the glomerulus is made up of three layers: the endothelial cells, a basal lamina, and a layer of cells with foot-like processes called podocytes. The endothelial cells have pores called fenestrae that allow fluid to pass through, whereas the podocytes loosely interlock to create slits in the cell layer. All three layers together create a filter that allows small molecules to pass through, but blocks the passage of large proteins in cells, leaving them in the blood. As a result, wastes such as urea, as well as water and key solutes such as electrolytes, glucose, and amino acids, all end up in the filtrate. After passing into the Bowman's space, the filtrate enters the renal tubule. The renal tubule of the mammalian kidney is divided into three sections: a proximal convoluted tubule, a loop of Henle, and a distal convoluted tubule. Although the renal tubule is one long tube, these three sections are specialized for different functions due to differences in permeability to different substances. After passing through the filtration barrier and into Bowman's space, the filtrate moves into the first portion of the renal tubule, called the proximal convoluted tubule. Here, electrolytes and other nutrients that the organism requires are reabsorbed into the blood, including glucose and amino acids filtered by the glomerulus, as well as most sodium and chloride ions. To maximize absorption, the proximal convoluted tubule has thick walls composed of epithelial cells with finger-like projections called microvilli on their surfaces, similar in structure to those that are found in the small intestine. The microvilli form a brush border along the lumen of the tubule, greatly increasing the cell's surface area for solute and water reabsorption. The epithelial cells possess numerous mitochondria that produce the ATP needed to actively reabsorb solutes from the filtrate into the bloodstream. Because the proximal tubule is permeable to water, water diffuses by osmosis in the same direction as the electrolytes and solutes. By the time the filtrate leaves the proximal convoluted tubule, about 75% of the water, along with most of the electrolytes and solutes, has been reabsorbed into the bloodstream. The proximal convoluted tubule connects to the loop of Henle, the second portion of the renal tubule. Although many nephrons contain short loops of Henle that are confined to the renal cortex, some descend all the way into the medulla before looping back up to the cortex. These longer loops of Henle play an important role. They create a concentration gradient in the interstitial fluid from the cortex to the medulla, with the interstitial fluid of the cortex being less concentrated and the interstitial fluid of the medulla being more concentrated. We can see this increasing solute concentration gradient with the arrow shown on the far right. This concentration gradient allows water passing through the downstream collecting duct to be reabsorbed, thus producing concentrated urine. The loop of Henle creates a concentration gradient because the two limbs of the loop of Henle run in parallel but in opposite directions, and they differ in their permeability to water and ability to actively transport electrolytes. Although the filtrate enters the descending loop before looping around to the ascending limb, it is easier to consider the ascending limb first. The thick portion of the ascending limb of the loop of Henle is impermeable to water, but actively transports electrolytes out of the filtrate. As the filtrate moves up the thick ascending loop of the loop of Henle, electrolytes are actively transported out of it into the interstitial fluid surrounding the loop of Henle. At the same time, the filtrate is moving from the descending limb to the ascending limb. As a result, the interstitial fluid becomes more concentrated, and the filtrate passing through the ascending limb becomes less concentrated. In contrast to the ascending limb, the descending limb is permeable to water. Therefore, water moves passively out of the descending limb, driven by osmosis. As water moves out of the limb, the filtrate becomes more and more concentrated, matching the concentration in the surrounding interstitial space lower in the inner medulla. As electrolytes are pumped out of the thick ascending limb, the filtrate becomes less and less concentrated. Although the filtrate enters and leaves the loop of Henle at about the same concentration, the trip around the loop creates a concentration gradient in the interstitial fluid surrounding the loop of Henle, with fluid in the inner medulla much more concentrated than that in the outer cortex. At the base of the loop, the filtrate still contains some electrolytes, but the main solute is urea. This is because electrolytes were actively reabsorbed into the bloodstream from the proximal tubule, and because the descending portion of the tubule is impermeable to urea. Then, as additional electrolytes are actively transported out of the thick ascending loop, the concentration of the filtrate, now mainly urea, decreases because the tubule is impermeable to water. In the loop of Henle, active transportation of electrolytes creates a concentration gradient, which is then multiplied because the descending and ascending loops move in parallel but in opposite directions. This system is therefore known as a countercurrent multiplier. Finally, the distal convoluted tubule is one of the main sites of secretion. Additional wastes not filtered by the glomerulus are actively secreted into the distal convoluted tubule from the bloodstream. The distal convoluted tubule also participates in the regulation of key electrolytes such as potassium, sodium, and calcium. It helps to regulate blood pH, in that bicarbonate ions are secreted into the tubule or reabsorbed out of it as needed. There is one more important point to consider. Typically, a high concentration of solutes in the interstitial fluid would be quickly lost due to water flowing out of blood vessels by osmosis. To avoid dissipating the concentration gradient, the blood vessels in the kidney are also arranged in a particular fashion. The blood vessels, collectively called the vasa recta, have descending and ascending vessels arranged in a countercurrent organization, just like the loop of Henle. The blood within these vessels flows down into the medulla in one arm and back up through the cortex in the other. This arrangement allows the concentration gradient to be maintained. Blood flowing from the cortex to the base of the kidney encounters increasingly concentrated interstitial fluid, allowing the passive diffusion of water out of the blood. However, the descending vessels pass in proximity to ascending vessels, which reabsorb the water. In effect, water passes straight from one arm of the vasa recta to the other, meaning the interstitial fluid and the bloodstream end up with no net gain or loss of water. As a result, the concentration gradient from the cortex to the medulla is maintained. The final segment of the nephron is the collecting duct. At this point, the key processes of filtration, reabsorption, and secretion have occurred. The filtrate is dilute but contains wastes and water. The collecting duct is the site where water levels are adjusted to meet the osmoregulatory needs of the organism and therefore maintain homeostasis. The concentration gradient from cortex to medulla, established by the loop of Henle, allows the collecting ducts to regulate water levels. The permeability of the collecting ducts to water determines how dilute or concentrated the urine is. The water permeability of the collecting ducts is controlled by the peptide hormone, antidiuretic hormone, or ADH, also called vasopressin. This hormone is secreted by the posterior pituitary gland. In the presence of ADH, the collecting duct walls become permeable to water. This change in permeability occurs because aquaporins insert in the membrane of cells of the collecting ducts. The aquaporins allow water in the collecting duct to diffuse into the interstitial fluid, thus making the urine in the tubule more concentrated. In the absence of ADH, the collecting duct walls are impermeable to water, preventing the water from leaving. This produces dilute urine. Diuretic substances such as caffeine and alcohol promote the dilution of urine by interfering with ADH receptors and therefore making the collecting duct walls more impermeable to water. If hypotonic urine is produced, there is net water loss. Consequently, diuretic drinks often cause dehydration. Because the kidneys receive a large fraction of the blood leaving the heart, they are well suited for monitoring changes in blood pressure and maintaining it at relatively constant levels, another example of homeostasis. Specialized cells of the efferent arteriole leaving the glomerulus of each nephron form the juxtaglomerular apparatus. In response to a drop in blood pressure, these cells secrete the hormone renin into the bloodstream. Renin stimulates epithelial cells of the lung to release an enzyme that converts an inactive hormone, angiotensinogen, in the bloodstream into its active form, angiotensin II. Angiotensin II acts on the smooth muscles of arterioles through the body, causing them to constrict, thus increasing blood pressure and directing more blood back to the heart. Angiotensin II also stimulates the release of aldosterone, a hormone produced by the adrenal glands that stimulates the distal convoluted tubule and collecting ducts to take up more salt and water. Increased salt and water uptake in turn leads to increased blood volume and therefore increased blood pressure. Thus, the kidneys participate in the control of blood pressure that is also monitored by baroreceptors, which are specialized mechanoreceptors that sense blood pressure in the aorta and carotid bodies. These communicate with the autonomic nervous system to control heart rate and contractile force. In summary, The kidneys play many important roles. In addition to the elimination of waste and other toxic compounds, they serve important homeostatic functions, including water and electrolyte balance, and the regulation of blood volume and blood pressure.